been around for probably 10, 15 years, but here in the United States, it's still not well integrated into medical care. And so I work with my company to make that happen because as Dr. Suhasini was talking about, precision and personalized medicine is the future of medicine. And when you can use your genetics to see the right strength and the right dose and the right medication for a patient, you can really optimize their therapy and decrease side effects. So as you can see on this slide, the goal of pharmacogenomics is to improve safety and effectiveness of a drug. People are very different and they respond to medications differently. And now that we have a look into their genetic profile, we can determine what drugs work for them, which ones do not, and an approximate dose, and therefore take most of the guesswork out of prescribing, which is wonderful. Here's a cover of a magazine from 2003, but it's still yet showing that this has been around for a while, but it's still yet to be integrated in personalized medicine. But I think as of this year, it's really starting to take off. So I'd like to talk with you today about what pharmacogenomics is for those who aren't familiar and how it can really help with personalized medicine. So here's an example of a population, all with the same diagnosis and the same prescription, but everyone responds differently. This is the group where the drug benefits the patient, but it's toxic, or it's toxic and not beneficial, or not toxic and beneficial, or not toxic, not beneficial. So everyone kind of falls into those 4 different categories for each medication, depending on their own genetic code. And so pharmacogenomics is neat in that it combines genetics with the pharmacology, and we can really get to precision medicine for our patients. The basis of pharmacogenetics is that everyone is different, even amongst family members, although we see that family members are significantly more similar than other people to have similar responses to medication, but even family members don't have identical responses to drugs. And especially, I appreciate the significant difference across ethnic backgrounds. Typically in drug studies, only one or few ethnic backgrounds are studied when the medication is studied in a patient. And here with the pharmacogenetic test, you're able to see how every person responds to it, and ethnic backgrounds can have a significantly different response to medications on whether they work better or worse, or more toxic or more side effects. And so this really helps to see if anyone in the world, how they're going to respond to medication, which is so invaluable. There's 3 different ways that the pharmacogenomics tests drug response. The first is pharmacokinetics, and this is how it changes the concentration at the drug site of the drug effect. And so these are our drug transporters, where it's transporting the drug across membranes. And the 4 main drugs, as you can see, are warfarin, clopidogrel, simvastatin, and metformin. And the next one is the pharmacodynamic change. This is the ability to influence at the target site, at the end response of where the cell is. And the 3 that are affected there are clopidogrel, simvastatin, and metoprolol. And you can notice that between these 2, metoprolol and clopidogrel both fall into both categories, which makes the difference in genetics even more complicated because patients may be a good responder in the pharmacokinetic aspect and a poor responder in the pharmacodynamic aspect. And so this helps to show how complicated our genetics can be, and with drug pathways and drug absorption, it can be influenced at all those different sites. And then the 3rd one is just underlying disease mechanisms, and these are not as obvious as pharmacokinetic or pharmacodynamic differences, but they also exist. And here's a picture. It helps to be a little more visual to see how this works in the body. So here's the compound, the drug we're looking at, and then we can see how it's absorbed, distributed, metabolized, or excreted. The gene affects all of that. So it can affect absorption, and if it's not even being absorbed to begin with, they're not going to have a good response. So that would be the patient would not want to go on something that they absorb poorly, or perhaps it deals with excretion. They absorb it, they distribute it, they metabolize it fine, but the patient has a problem excreting it, so it builds up to toxic levels in their body. So when that happens, we see poor drug responses with increased side effects and increased toxicity if our body can't get rid of it. So all of these different areas are affected by our genetics. And so we get our concentration of the drug, and then the concentration can go up or down, and then we still have to go to the cell and the receptor. And this is another place where the genetics affect the action of the drug on the cell. So it's a very complicated chain, but for individual patients, we can see how they can best absorb and excrete these medications. This kind of says the same thing. I'll move along. So pharmacogenomics uses the approaches to see the differences in people. What happens on the DNA level is a single nucleotide polymorphism, or it's abbreviated as a SNP. These are where the changes are in the DNA that marks us different in responding to the medications. And there's more than 1.4 million of these in the genome, with 60,000 in the coding region. And so that shows on the bottom of this slide how, even though people are 99.9% similar in their genetics, that 0.1% difference makes a very big difference in our response to medications. And luckily, now that we have sequenced this and we know how each response goes, we can prescribe appropriately. And there's a international consortium called CPIC, and they have summarized all of these findings and help guide the prescribing. So you find out how each patient responds to each pathway, and then can prescribe accordingly. So here's all the different mutations that can happen to yield a different response to medication. And I won't go into detail and bore you on all of those, but this is just showing that there's quite a few different ways that our genetic differences yield different responses to medication. And that's called a polymorphism. And so here's the image of the DNA and that SNP happening, where a single base pair is changed, and how once that's changed, then the response to medication is different. And in pharmacy, we know about the cytochrome enzymes where we metabolize our drug. There's 15 known to metabolize them, and 7 have specific polymorphisms. And I'd like to talk with you about those now. You can see here how everyone has the 2 alleles in their DNA. And if you have 2 functional alleles or increased activity alleles, then you are going to be an ultra rapid metabolizer, which means that you metabolize it so quickly, you're likely not getting the benefit from this medication. It's going through your body too quickly, and you don't get a therapeutic effect. The second one is 2 wild type or normal alleles, and this is categorized as an extensive metabolizer, which is a normal capacity. This is where we want our patients to be. They want to be an extensive metabolizer that gets the appropriate dose without getting too many side effects. And these 2 down here are intermediate or poor metabolizers, because they have either 1 defective allele or 2 defective alleles. And when that happens, they have a reduced capacity. They may absorb it just fine, but they're not metabolizing it quickly enough to get it out of their system. And so in these 2 categories, it builds up to quite high levels in their body. And when this happens, you see the significant side effects and the toxicities, because this medication is building up to high levels in their body and they're not excreting it well. And this is a slide that kind of shows what we just talked about, how the variance can affect the success significantly. So I'll just share with you one example, and then we'll move to questions, because I know I'm the last speaker of the day and I don't want to keep you long. But my example is in cardiology with the medication clopidogrel. It's a thienopyridine, and this one is my poster child for the importance of pharmacogenomic testing in patients, because clopidogrel is a prodrug. So it requires hepatic bioactivation to be active in the body. It's not active until it gets metabolized first. And let me move down here. Yeah, so clopidogrel must be activated first before it can work in the body. However, 30% of people metabolize this too quickly, and so it doesn't get to an appropriate concentration to prevent platelet aggregation. And so this graph shows it really well. So 30%, this whole four bars on the left show that patients are not reaching a therapeutic level on the clopidogrel. And that is extremely significant, because after you have an MI incident and someone goes on clopidogrel to prevent another MI or a stroke, all of these people, 31% of people, are not getting an appropriate level. So it's almost like they're taking a sugar pill. And so to consider the cost and the problem it would be for these people to be taking clopidogrel thinking it works, when in fact it does not, and they're at an extremely high risk for another adverse event, and with a significant cost or even a death after a second MI if the clopidogrel is not working. So this is the example of medications I use when I talk with prescribers about the importance of pharmacogenomics. Because if a drug is not even working at all for a patient, that is a very big problem, especially for something like clopidogrel that's keeping them alive. So this, I love clopidogrel for that example. And then with just a test, you could see this works or it doesn't. And if you're one of the ultra rapid metabolizers, then you don't even take clopidogrel, you use a different medication completely. So I appreciate this because it helps show what works and what doesn't. It takes the guesswork out of prescribing, and especially with the mental health medications. I won't go into that now because that's a whole other discussion in itself, but with mental health medications, it also takes the guesswork out of prescribing, whereas right now doctors are just guessing and patients are on years-long trials, increasing doses, adding new therapies, waiting for the drugs to become effective, and they're suffering the whole time. Whereas with a simple genetic test, it's non-invasive, it's just a cheek swab or a spit sample. So it is very simple to collect, non-invasive, and then for the rest of their life, they'll know how doctors should be prescribing for them because your genetics do not change. So it's nice in that you do this test once, and for the rest of your life, patients can have the therapy that is best serving them. So thank you for your time today. I love talking about pharmacogenomics, and if you have any questions, I'd love to answer those for you. It's been 15 years that this technology has been around, but that's the biggest hurdle. It used to be cost is the biggest hurdle, but now it's down to a very reasonable level. And so the biggest hurdle is getting prescribers to be aware of this and to use it. Luckily, what I do is I target medical practices that can benefit the most. So I specifically target the cardiology doctors as well as the mental health doctors, which is where patients see the most benefit from it. But just having those discussions and seeing if doctors are even aware that this technology exists, because so many of them don't even know. They just don't even know it's an option. However, the results are so significant, especially with the clopidogrel example, that there's started to be litigation against doctors if they're not using it in the United States in recent years, when patients come back and say, why didn't you test me? All it took was a test. Why didn't you even test me? So exactly. Unfortunately, it's not the ideal way, but because of those instances where patients are are fighting back that it's a combination that I think that's starting to become more of a norm. But this is our pharmacy space. This is what pharmacists do. It's not necessarily up to the doctors to champion this as much as us, because it's our space with pharmacology and pharmacodynamics. So it's definitely has room for potential in pharmacy, and how pharmacists are going to champion this new future, because this is the future of pharmacy. This is a huge part of it.
Yeah, because with pharmacogenomics now and this personalized medication, is actually becoming apparent that some of the wrong prescribing that the doctors actually do can be minimized or even eradicated altogether. And therefore, you're getting better treatments or better drugs for patient treatment. And also, it will be on the personal level. And as you correctly said, they maybe by pharmacokinetics may be fantastic, but the pharmacodynamics may not be up to scratch. Or maybe everything is okay, but just that kidneys is not doing the job that it's supposed to do. And therefore, we need to find a way to actually train the doctors so that they actually are on the same level with us, and saying that, yes, I may be thinking of X, but no, I'm dealing with Y. So what is Y's personalized prescription that I can give? And also, with all the talks that we've had today, everything really, really wraps together that we need, because a patient may be, maybe 5 milligram clopidogrel may not be adequate, or it may be too much for a patient. Need to really reduce it. But how do we know how to reduce it? We need to go to this pharmacogenomics so that we actually know the details about each individual patient. And that first slide that you had on, I actually had it on for one of my students, so that actually teach them pharmacogenomics, which is absolutely
good.
fantastic. So well done.
Oh, well, thank you. I'm glad you're here, and that there's a lot of pharmacists here, because this is the future of pharmacy. And as we champion it and move it forward, we can do great things.
Okay.
Best